Welcome to the Longevity Papers. This is the analysis where we take the week's most important scientific output and distill it down to what you, the researcher, investor, or just a deeply curious individual, absolutely need to know.
It's great to be back. And you're right, this week's sources weren't just a handful of interesting experiments. This was a period that presented a cohort of papers that are, genuinely model-changing.
Absolutely.
They challenge assumptions that have been foundational to aging biology for decades. Really core stuff.
Exactly. Our mission today is focused entirely on those model changes. We're looking at sources published roughly between, December 1st and 7th, 2025. And we're prioritizing work with really strong institutional backing and, just genuinely surprising results that require us to reshape our understanding of these fundamental mechanisms.
Right. We're skipping the incremental progress this week.
Yeah, we're focusing on the revolution.
And we have 5 major papers. They span these, highly conserved signaling pathways like ICE. They challenge classic evolutionary trade-offs. They provide these high-resolution human biomarker maps, dissect genetic architecture.
And even offer a blueprint for synergistic interventions.
Exactly. So if you are out there developing a drug, designing a diagnostic, or allocating R&D capital, this information is critical.
Okay, let's unpack this, starting with a finding that, it fundamentally rewrites the textbook on what we thought was maybe the most highly conserved and best understood longevity pathway.
Insulin IGF-1 signaling, ISIS.
Let's get into it. This first paper, a 4-fold male-specific lifespan extension via canonical insulin GF1 signaling. It comes from Russell, Lynn, Lasher, and colleagues at the University of Alabama at Birmingham.
And it was posted on bioRxiv, that's bioRxiv, on December 5th.
And to really appreciate the scale of this finding, we have to start with the foundational context, right?
We absolutely do. Since the early 1990s, the entire field has operated on this single, powerful paradigm. It was established by Cynthia Kenyon's groundbreaking work. The DAF2 mutation, specifically the E1370 allele, was the first real proof of concept.
The first proof that aging was plastic.
Exactly. That it could be controlled by a single gene. It was a monumental discovery.
And that finding established the gold standard, that reducing IS, the body's nutrient sensor pathway, roughly doubles the lifespan of C. elegans hermaphrodites.
Right. Doubling lifespan from about 20 days to 40 days. That became the canonical benchmark.
Yeah.
It's what every other intervention was measured against. It's immutable textbook knowledge.
Or it was.
It was mutable until Russell's team came along. They took the exact same fundamental genetic perturbation, the DAF2 mutation,
The very same one.
and applied it to male C. elegans. And the result is, it's astonishing. Instead of a doubling of lifespan, they achieved a 4-fold extension of lifespan. We pushed these males to live beyond 110 days.
Hang on, let's process that for a moment. 4 times the normal lifespan.
4 times.
That's an order of magnitude increase in potential, simply by changing the sex of the organism that's undergoing the intervention.
It absolutely mandates a shift in our thinking about ISIS. We can no longer view sex as some kind of tertiary or, confounding variable that causes slight variations in results.
Right, it's not just statistical noise.
Not at all. This study establishes sex as a primary determinant of longevity potential. It's capable of amplifying the output of a core, conserved aging pathway to an astronomical degree.
The numbers are just. If the maximum potential lifespan from the most well-known aging mutation can be amplified by 200%, from 2x to 4x based purely on sex,
then it tells us we fundamentally misunderstood the interaction between sex-specific biology and metabolic signaling. We've been missing half the story.
I am curious, though, about the methodological rigor. Finding this big, you have to be skeptical. Did they confirm that the DAF2 mechanism in the males was indeed canonical, the same pathway?
That's a key detail, and they addressed it in the source. They performed really rigorous controls to ensure that the ISIS pathway was reduced via the same canonical signaling cascade.
So, through DF16-FXO.
Precisely. Through the F16-FXO transcription factor in both sexes. The difference is not in the input signal, the DAF2 mutation is identical, but in the output leverage that's provided by the male physiology.
So it's not a different mechanism, it's just a more powerful response.
Exactly. And to your point about other factors, they even decoupled the longevity effect from reproductive strategy. They showed that even sterile males retain the full 4x extension. So it's not about mating stress or anything like that.
And crucially, this extreme longevity isn't just about extended survival. The source explicitly notes this 4x extension is coupled with a dramatic extension of health span.
Which is the whole point, really.
It has to be. They measured parameters like movement and feeding behavior, right? So this is a genuine rejuvenation signal, not just, extended frailty.
Absolutely. The metabolic state achieved by the low ISIS male is fundamentally healthier for much, much longer.
The therapeutic implications of this for any company working in the ISIS space, which is basically everyone, given how central nutrient signaling is, are just enormous.
Think of the clinical pipeline right now. You have GLP-1 agonists like semaglutide, you have metformin, even mTOR inhibitors like rapamycin. They all intersect deeply with nutrient sensing.
All of them.
So if a canonical ISA reduction strategy works 4 times better in males than in the standard hermaphrodite model, then future clinical trial design must immediately become sex-specific. It's not optional anymore.
That is a completely game-changing consequence. It raises this very real possibility that if current or, past ICE-lowering interventions showed only modest overall success in mixed-sex human trials, it might have been because this massive effect we're seeing in the male cohort was just diluted by a less dramatic, maybe only 2x equivalent effect in the female cohort.
You could have a blockbuster drug for 1/2 of the population just hiding in plain sight.
Buried in the average.
Exactly. So we need to dissect the why. What is driving this profound sexual dimorphism? The source analysis offers a few highly plausible, untestable hypotheses.
Okay, what are they?
First, differential testosterone-insulin signaling crosstalk. We know that androgens, specifically testosterone, modulate insulin sensitivity and glucose uptake.
So the idea is that the radical metabolic shift from the DAF2 mutation might synergize in a unique way with the male hormonal environment.
Right. It could provide protection against typical age-related metabolic decline that the female environment either doesn't require in the same way or simply doesn't amplify.
Okay, that's one. What's the second possibility?
Alternatively, we have to consider fundamental differences in what's called mitochondrial sexual dimorphism. Males and females often exhibit differences in mitochondrial function, oxidative stress tolerance, energy flux.
So the starting point is different.
The starting point is different. If male mitochondria are, let's say by default, operating less efficiently or maybe closer to a critical stress threshold than female mitochondria, then the profound metabolic optimization you get from the DAF2 mutation might offer them a disproportionately larger survival advantage.
It's like installing a new clean engine in a car that desperately needed it versus one that was already running fairly well.
That's a perfect analogy.
And
the third hypothesis involves the existence of sex-specific metabolic checkpoints that are regulated by ICE.
so pathways that are just there in one sex but not the other.
Or are regulated differently. The source suggests we need a transcriptomic comparison, a molecular readout of which genes are switched on or off in male versus female DAF2 mutants. We need to find where the pathways diverge at a molecular level. Are there unique metabolic enzymes or repair pathways that are only massively upregulated in the male when ICE is low?
For the listener, then, the needed follow-up is clear. And it's expensive. You need hormonal dependency testing manipulating androgen and estrogen levels in both sexes under the DAF2 regime to establish causality.
That's step one.
And most importantly, you have to confirm if this mechanism translates to mammalian models. We must replicate this in sex-stratified studies of GHR knockout mice, which already show profound longevity.
And if a synergistic effect is seen there, even if it's just a 2x amplification difference between sexes in mice, the R&D strategy for ICE-targeting drugs changes overnight, globally.
The potential is just staggering. Now, while this paper focuses on maximizing somatic lifespan via ISIS, it implicitly raises that old nagging question: what's the cost?
The trade-off.
Does maximizing somatic health always come at the expense of reproductive fitness? The next paper tackles this trade-off directly.
So the second paper, published on the same day, December 5th, also on Bioxforce, immediately links back to ISA, but it addresses a really foundational concept in evolutionary biology. It's from Duxbury, Godin, and colleagues from the University of East Anglia.
The title is Lifespan Extending Downregulation of Insulin Signaling Reduces Germline Mutation Load.
And this goes right at the long-standing dogma of the somatic germline trade-off. For decades, evolutionary theory, which is really encapsulated in the disposable soma theory, dictated that organisms have limited resources.
Right. To survive, you have to allocate those resources efficiently. So, investing heavily in somatic maintenance, your body's health and longevity, was thought to necessarily compromise your reproductive fitness.
Or the integrity of your germline, the cells responsible for the next generation. It made perfect evolutionary sense. Why invest enormous energy into maintaining your body indefinitely if you've already successfully passed on your genes?
The cost of reproduction was high, and the maintenance cost was the price you paid for a long life.
And we saw this reinforced everywhere. Most interventions that extend lifespan, like caloric restriction or strong mTOR inhibition, they often dramatically reduce fecundity. They reinforce this model.
And this study,
Yeah.
it challenges that fundamentally.
It does. The stunning finding is that ice reduction doesn't force this trade-off. Instead, it seems to create a state of whole organism genomic homeostasis,
Improving both at the same time.
simultaneously improving somatic lifespan and germline genome integrity. So the system isn't just diverting limited resources from one bucket to another, it's operating with vastly superior overall efficiency.
And how did they test this?
They induce this longevity later in life using adult-onset ISA downregulation with DAF2 RNAi. That confirms it was an active, adult-driven mechanism, not just some inherited trait from development.
Okay, and the key finding?
The quantifiable key finding is remarkable. They reduced germline single nucleotide mutation rates by up to 50%.
50%?
50. And crucially, this protection occurred without any detectable cost of fecundity or lineage persistence across multiple generations.
A 50% reduction in germline mutation load is colossal. That level of protection means reduced ISIS isn't just promoting cellular housekeeping in the somatic tissue.
No, it's actively protecting the core genetic blueprint against internal damage and external stressors. They specifically showed it prevented the expected elevation of mutation rates that's induced by UV exposure.
So it's a proactive defense system.
It is. And this elevates ICE far beyond a simple somatic aging regulator. It positions it as a central coordinator of whole organism genomic stability. And it does it via an epigenetic pathway.
And we know the mechanism. The source points out that this genome protective effect critically requires a specific protein.
The germline nuclear argonaute, HRDE1.
Let's delve into HRDE1 because for the deeply curious listener, this is where the mechanism gets really interesting. What is this argonaute pathway actually doing here?
Okay, so think of the genome as this vast library. It contains not just helpful textbooks, which are the genes, but also thousands of genomic saboteurs, these things called transposable elements or jumping genes.
Right, and these elements are notorious drivers of genomic instability and mutation when they jump around.
Exactly. The HRDE1 argonaute protein acts as an epigenetic quality control team. It uses small RNA molecules as a blueprint to identify and target these transposable elements and other mutation-prone loci, and it actively silences them through methylation and other epigenetic marks.
So ICE reduction is the signal that tells the organism, okay, resources are low, efficiency must be paramount.
Right.
And the HRDE1 pathway is the mechanism that responds by sending out this epigenetic cleaning crew to silence genomic instability, which suggests it requires active epigenetic regulation, not just the passive benefits of a slowed metabolism.
The synthesis is so powerful. ICE is not simply slowing metabolic output. It's inducing a high-fidelity state of genomic maintenance.
And for human therapeutics, this has implications that stretch beyond individual lifespan, right? This could go into public health.
Absolutely. If this mechanism is conserved, ICE-lowering therapies could potentially protect not only your somatic tissues, reducing cancer, reducing neurodegeneration, but also your germ cells.
Which could mean protection against heritable mutation accumulation, essentially improving the health trajectory of your descendants.
You got it. This is a major biological hurdle that's potentially bypassed.
Okay.
The big critique against radical lifespan extension often centers on this idea that we would just create longer living but weaker lineages.
Right, that we'd live longer but pass on more accumulated damage.
But Duxbury's work suggests that through optimized ICE reduction, we can achieve longevity while simultaneously reducing heritable disease risk.
Okay, but productive skepticism is needed here. The trade-off theory is resilient for a reason.
Of course.
The source must note the need for follow-up. At what threshold of ICE reduction does this germline protection emerge? If it requires such a deep, resource-expensive reduction that it severely impacts overall population size, or requires massive shifts in resource allocation?
Then the trade-off might still exist, just shift it. That's a fair point.
Yeah.
But if you can get 50% protection with a mild, clinically tolerable ICE reduction,
That's an incredibly high-value proposition.
it is. So the next steps for research are clear. We need to test the HRDE1 homologs in mammals, specifically the P-will proteins, to see if an equivalent germline protection occurs with ICE's reduction in mice.
And since the potential benefit is heritable, multi-generational mammalian studies are non-negotiable. We have to confirm that these protective epigenetic benefits are genuinely passed down.
Right. So we've seen how reducing ICE can protect the genome, establishing new targets and challenging these old evolutionary models. But how do we measure this state of enhanced health span and aging trajectory in actual humans?
We need a better diagnostic that integrates all these metabolic changes. Shifting our focus entirely now, let's turn to high-resolution human data and biomarkers. Our third paper is a global metabolomic and lipidomic landscape of human plasma across the lifespan.
This is from Xinru Liu, Tingting Liang, and colleagues at the University of Science and Technology of China. It was published in Aging Cell on December 6, 2025.
This is a massive foundational effort. It's providing one of the most comprehensive metabolic maps of human aging to date.
It really is. They employed untargeted metabolomics and lipidomics, profiling on plasma samples from 136 individuals, spanning from birth all the way up to 84 years. So this isn't just a snapshot. It's a high-definition time-lapse of metabolic drift across the human life course.
We already have dozens of aging clocks, though, mostly based on DNA methylation, like Grimmage or Dundon pace or proteomics. So what's the true novelty here? The model change.
It's not just what they measured, but how the data changed over time. That's the key.
Okay.
The most critical finding regarding the dynamics of aging is the nonlinear metabolic trajectory. They found that aging is not a continuous linear drift where everything slowly degrades at a constant pace.
So it's not a gentle slope.
No. Instead, the analysis reveals discrete, critical transition points, metabolic cliffs, if you will, where the body undergoes punctuated metabolic reprogramming at specific life stages. This suggests the aging process has these scheduled shifts in chemistry.
Which offers defined windows for intervention.
Precisely. That concept of punctuated change is powerful. It means the vulnerability landscape changes throughout life.
Let's talk signatures. What specific metabolites characterize the beginning and the end of this spectrum?
The extremes really tell the story of resource allocation. Newborns display these unique profiles dominated by certain sphingosine, SPH, species.
Which makes perfect evolutionary sense. SPH plays a critical role in cell membrane signaling and the massive demand for myelination and neural development early in life.
The body is focused on building and insulating the brain. That's its priority.
And conversely, the aging signature, what does that look like?
It's characterized by two really distinct features: elevated amino acid levels and extensive lipid imbalances.
The elevated amino acids, we're talking branched-chain amino acids, DCAAs, and aromatic amino acids. That's a hallmark of metabolic dysfunction, right? It often signals impaired protein turnover.
Or, more likely, mitochondrial failure to efficiently process these compounds for energy. They just accumulate in the plasma because the utilization pathways are overwhelmed or compromised. They can't burn them fast enough.
And the lipid imbalances are particularly concerning. We're moving beyond just general fat accumulation to specific pro-aging lipids.
That's right. The shift points to significant mitochondrial dysfunction and a loss of membrane integrity. They specifically highlighted the accumulation of highly pathogenic lipids like ceramides and various species of oxidized lipids.
And we know ceramides are heavily implicated in driving insulin resistance, cellular senescence, apoptosis.
All the bad stuff.
Yeah.
This data reinforces the idea that lipid metabolism, particularly the accumulation of these detrimental species, is a core genetically programmed driver of aging. It's not just a downstream consequence of metabolic disease.
So the shift from an SPH-rich brain-building profile in newborns to this ceramide and amino acid disbalance in older adults it's the decline of function written directly in the blood plasma.
Exactly.
Yeah.
Now, they developed an aging clock based on this metabolite profiling. For the researcher, the immediate and most skeptical question is, does this new clock outperform existing validated epigenetic clocks?
Right. Does it beat Grimmage or Dundon pace in predicting all-cause mortality or morbidity? The source provides the map, but we need predictive power.
And that remains the critical question. It requires immediate head-to-head validation. But the strength of this metabolite clock is unequivocally its clinical practicality.
It's based on plasma.
Exactly. Which means rapid, cheap, and easily deployable profiling in standard clinical settings. It's much simpler than methylation clocks, which require specialized infrastructure and tissue prep.
So even if it provides slightly less predictive variance than the best methylation clocks, its accessibility means it can be applied to massive clinical cohorts immediately.
Its value really hinges on its ability to provide actionable, reversible metabolic targets, and that leads to the therapeutic leads.
If amino acid disbalance is such a feature of aging, then targeted intervention using amino acid supplementation, maybe modulating the balance of BCAAs or increasing glycine, becomes a rational, data-driven drug target.
Likewise, interventions aimed at lipid remodeling. Anything that targets ceramide synthesis or degradation pathways or supplements things like plasmalogens are highly rationalized by this extensive human data.
And the identification of those discrete metabolic transition points gives us optimal therapeutic windows. If the data suggests a critical metabolic cliff occurs around age 45, a point they identify, that becomes the prime time for preventative intervention to maximize returns.
Before the accelerated metabolic drift takes hold.
So the necessary follow-up here isn't just validating the clock against scrimmage. We need to pair this plasma data with tissue-specific metabolomics at those identified transition points.
We need to know which tissue, liver, muscle, adipose, brain is driving the metabolic reprogramming. That will define the target organ for drug delivery and ensure precision intervention. Okay, moving from the metabolic phenotype to the underlying genetic architecture. Our fourth paper is Genome-Wide Association Study of Proteomic Aging Reveals Shared Genetic Architectures with Longevity, Early Life Development, and Age-Related Diseases.
This one is from Arjun Thierry, Lofton, and the team at Massachusetts General Hospital. It was posted on Medarich on December 4th.
And this paper, I think, provides the second major model change this week. It moves proteomic aging, the measure of misfolded or poorly functioning proteins, from just being a late-life biomarker.
To being a genetically heritable, causally relevant determinant of lifespan.
Precisely. They conducted a massive genome-wide association study, a GWS, on the proteomic aging gap, or protage gap, in the UK Biobank. It involved almost 39,000 individuals.
And the definition of protage gap is key here. It's the difference between your predicted age based on your plasma proteome and your actual chronological age.
Right. A positive gap means you're aging proteomically faster than you should be. The core finding linking genetics to this measure demands absolute attention. They identified 27 distinct genetic loci associated with protage gap.
27 specific locations in the genome, but the truly staggering number is the genetic correlation. Protage gap shows a genetic correlation with exceptional longevity of RG equals 0.83.
In genetic epidemiology, an R value that strong, it's almost unheard of, where a negative gap aging slower is almost perfectly correlated with a long life. This suggests it's far beyond simple association.
It implies a near-direct, shared underlying genetic determination.
The genetic factors that accelerate your proteomic aging are the same factors that shorten your life. They then leverage this by creating a protage gap polygenic score, a PGS, based on those 27 loci.
And the predictive power of that PGS was then validated in a huge cohort, over 500,000 individuals in the FinGen Biobank.
And it held up. It showed that a favorable protage gap PGS was associated with a 1.43 increased odds of achieving exceptional longevity.
So we have a genetically driven tool that can identify individuals who are predisposed to accelerated aging, and it's strongly linked to their overall lifespan outcome. But the most profound conceptual insight, the biggest model change this paper delivers, is this life course model they uncover.
This is what I found deeply fascinating. The idea that aging is pre-programmed somehow, tying late-life health to markers from early life.
Exactly. They showed that the same set of proteomic aging genes that predicts late-life disease, risk obesity, cardiometabolic disease, osteoarthritis, also affects early developmental timing in children.
How did they show that?
They used data from the Adolescent Brain Cognitive Development, or ABCD, study, and they correlated these protage gap genes with factors like skeletal maturation and early cognitive development.
It's unsettling to think that the speed at which you matured, or the rate your skeletal system developed as a kid, might be genetically coded to predict your risk of a hip replacement decades later because of osteoarthritis.
It connects the dots from the cradle to the grave in a terrifyingly direct, genetically determined way.
It implies there's a slow development, slow aging genotype versus a fast development, fast aging trajectory. And it's programmed before you even hit puberty.
And they didn't stop at the correlation. They provided a specific molecular mechanism for this life course integration. Colocalization analysis identified the FTO gene,
Which is known primarily as a major obesity and metabolic risk gene. But here it's identified as a key player uniting all these diverse aging traits.
Why is FTO so powerful in this context, though? beyond just its link to obesity?
Well, FTO doesn't just regulate fat storage. Its primary molecular function is as an RNA demethylase. It regulates something called M6A methylation of messenger RNAs.
Okay, so think of M6A methylation as a tag that controls the stability and the speed of gene expression.
Exactly. So by affecting FTO function, you are essentially accelerating or decelerating the reading of the body's entire instruction manual. This implies that the dysregulated metabolic pathways and the accelerated proteomic aging they observed stem from a fundamental genetically driven alteration in the control of gene expression kinetics via M6A.
That's a fundamental switch. For therapeutic development, those 27 identified loci are instantly high-value targets. You're addressing the genetic roots of aging, not just the symptoms.
For sure.
But let me introduce some productive skepticism here. A 1.43 odds ratio is compelling, but how much variance does that PRODEG gap PGS actually explain compared to, say, environmental factors or lifestyle?
That's the right question.
Are we chasing a powerful genetic signal or one that's easily overcome by healthy living?
And that's fair. While the correlation, the Rorschach of -0.83, 0.83, suggests causality is strong, the predictive power of any PGS is always moderated by environmental factors.
Okay.
But the source's value isn't necessarily that the PGS is a perfect crystal ball.
So what is it?
It's that it identifies a genetically susceptible population. The immediate utility is for stratifying future intervention trials. If a biotech develops a new metabolic therapy, they should test it, stratifying by the PRODEC PGS.
So individuals identified as genetically predisposed fast agers could be targeted for preventative therapies years or even decades before the onset of symptomatic disease.
This is the definition of precision aging medicine.
It's the gold standard for GWAS then. Moving from statistical association to a defined genetic architecture with 27 loci, to a specific causal mechanism in FTM-M6A, and finally, to immediate clinical utility with the PGS.
It's a very complete story. We need functional validation of the 27 loci and mechanistic dissection of the FTM6A regulatory function in tissue-specific models immediately.
Absolutely. So we've explored genetic predisposition and deep metabolic biomarkers. Our final paper for the week moves us toward actionable, multimodal intervention strategies, providing a concrete blueprint for how to use this knowledge. Our fifth and final paper takes us into the realm of complex synergy. This work is titled Gut Metabolism Links Precision Nutrition, Exercise, and Health Span in Drosophila melanogaster.
This is from Wei, Liu, Sun, and colleagues at Duke University School of Medicine, posted on BioArc4 on December 2nd.
And this paper provides a direct challenge to the fundamental cost of living hypothesis.
As we discussed earlier, traditional lifespan extension methods, caloric restriction, strong IC inhibition, they often come at the severe cost of reproductive capacity and physiological performance.
You see reduced fertility or poor movement, what they call locomotor function. It's the trade-off in action.
Right. And the Duke team found a way to uncouple that trade-off. They achieved what is really the holy grail of maximizing lifespan without sacrificing physiological function.
And how did they do it?
They used a synergistic, multimodal intervention. It combined three specific and, importantly, highly implementable components: methionine restriction, MR, taurine supplementation, tau, and moderate exercise.
The synergy is critical here.
Absolutely critical. When they applied all three simultaneously, they achieved synergistic lifespan extension while managing to preserve reproductive capacity and demonstrably improving locomotor function.
Which is exceptionally rare.
It is. It suggests that this combination engages a fundamentally different metabolic mechanism, one that is highly efficient and optimized, rather than just slowed down or starved.
Let's delve into that mechanism because it's counterintuitive to the traditional thinking about restriction. If you're extending life, shouldn't metabolism slow down? They found the opposite.
Correct. Integrative metabolic tracing showed a central feature of the long-lived, high health span flies was increased mitochondrial TCA cycle flux in the gut.
The tricarboxylic acid cycle, the Krebs cycle, that's the core engine room for energy production.
Right. So instead of slowing down metabolism to conserve resources, which is the traditional CR model, the system is optimized for clean, hyper-efficient energy production and utilization, specifically in the gut tissue itself.
But why the gut? Why is that the focus?
The gut is the interface between the outside world nutrition environment and the host. Optimizing TCA flux here means that nutrient extraction and processing are maximally efficient, minimizing metabolic waste and energy deficit.
Okay, so it's a more efficient engine.
A more efficient engine that's coupled with demonstrably enhanced redox homeostasis, which just means better antioxidant defenses, and reduced oxidative stress.
Mhm.
It's the difference between a high-performance engine that's running rough and polluting and one that is finely tuned, running cleaner, and lasting longer.
And what coordinates this complex, optimized state, linking the diet and
The
exercise?
gut microbiome.
Of course.
Absolutely. Lactobacillus plantarum was identified as a key microbial mediator. This specific bacterium was highly responsive to the dietary and behavioral stimuli. This suggests it's not just a passive member of the ecosystem,
But an active conductor.
an active conductor, coordinating host energy metabolism. It's likely achieving this by producing signaling molecules that directly affect the host's intestinal cells and mitochondrial function.
So the synergy makes perfect mechanistic sense then. Methionine restriction initiates the nutrient-sensing signaling cascades like mTOR and AMPK.
Right.
Taurine, which is known to protect mitochondria, supports that enhanced redox balance needed for high performance. Exercise builds metabolic capacity.
And Lactobacillus plantarum acts as the control node, coordinating the communication between the diet and the host's gut engine.
And if you break any one link,
It falls apart. Removing the exercise or removing the taurine significantly reduced the maximal synergistic benefit. This is a powerful validation of a multimodal approach to aging. It proves the effect is greater than the sum of its parts.
the implications for human health span strategies are profound because all three of those components are highly translatable and implementable at scale.
They really are. Methionine restriction is achievable through precision dietary choices. Taurine is a readily available, safe supplement, and moderate exercise is the standard of healthcare.
This gives us a concrete, validated framework for multimodal health-preserving interventions.
The necessary follow-up here is immediate. We need to test this exact combination in mammalian models, specifically mice, to see if the lifespan extension, the reproductive preservation, and the vigor are conserved.
And furthermore, we have to determine causality. You have to knock out L. plantarum or suppress it to see if the benefit persists.
If the maximal benefit disappears, we know the microbe is causally required for this synergistic effect. It's not just a bystander.
And finally, identifying the specific bacterial metabolites that mediate this effect. Is it butyrate? Is it another short-chain fatty acid or some novel molecule that would then provide a target for synthetic biology or precision probiotic design?
Moving us beyond simple supplementation to targeted microbial manipulation.
Okay, let's unpack this incredible week of sources and synthesize the 5 core model changes we've seen. We started with the foundational pathway, IC, and we learned via Russell's work that our understanding was critically incomplete. IC longevity amplification is dramatically sex-dependent.
Leading to a 4-fold extension in males. Just incredible.
We immediately followed that by addressing the most persistent evolutionary cost, with Duxbury showing that the somatic germline trade-off might be entirely avoidable.
Right, via ICES reduction paired with that HRDE1 epigenetic mechanism, which actively protects the germline genome from mutation. That is a fundamental rewriting of evolutionary resource allocation theory.
Then we shifted to deep human data. Lou's metabolomics revealed that aging is not a smooth process. It's punctuated by these discrete, nonlinear metabolic transition points.
Characterized by amino acid and ceramide disbalance.
And Argentieri's genetic work provided high-value therapeutic targets, 27 loci, and a staggering genetic correlation, that ARG of 0.83, linking proteomic aging to lifespan.
And crucially to early life developmental timing via the FTOM6A pathway.
We saw a strong concerted move this week from just correlation to real mechanism and causality.
Absolutely. The 2 ISA papers provide deep, actionable targets, sexual dimorphism and the HRDE1 epigenetic control pathway. The 2 human papers give us urgently needed actionable biomarkers and high-value genetic loci for stratified trials.
And the Drosophila work, by way, gives us a proven blueprint for a synergistic health-preserving intervention that bypasses the traditional reproductive cost.
This knowledge is just. It's vital for any biotech researcher designing the next generation of trials. If you are developing ice-lowering drugs, you now have to think about sex-specific dosage and primary outcome endpoints.
You have to stratify.
You must stratify participants by sex, by metabolic profile, and by genetic risk if you want to capture the full potential effect.
And if you're building a diagnostic, the task is now clear. You have to test the power of proteomic and metabolic clocks head-to-head against methylation clocks to see which best captures the integrated genetic and environmental drivers of the aging process.
Especially at those crucial transition points that were identified in the metabolic map.
So if the Argentieri paper is correct, that our proteomic aging trajectory is strongly correlated with early developmental timing, and it's set by genetic switches like FTO. It raises an important and, I think, provocative question that we should leave you with to mull over.
And that is, are we intervening too late? Does true longevity protection require optimizing the metabolic and genetic landscape decades before the first signs of age-related disease appear?
If the trajectory is indeed set in childhood, then perhaps the most effective longevity intervention programs will be those focused on early developmental optimization.
Shifting the paradigm from treating the aged to optimizing the young and the genetically susceptible. Something for you to explore on your own as you process this data.
Thank you for joining us for this week's analysis. We hope this exploration into the source material was valuable for your research and your understanding.